Good afternoon, everyone, or good morning for those of you that are listening from a different time zone. Welcome to the first webinar of the Virtuous Massive Open Online course, Promoting Ocean Literacy and Project-Based Learning by Monitoring Biodiversity. I am Tekla from Submon, one of the partners of Virtuous project, and I would like to introduce you today's presenter, Susan Gottensparre and Michael Holsson. Hi, Susan, and hi, Michael. They are from the University of Gothenburg, Sweden, and they are the coordinators of the Erasmus Plus funded project Virtuous. And today we will have Adam Frederick. Hi, Adam. Hello. From the University of Maryland Center for Environmental Science and Maryland Sea Grant in USA. Adam is an assistant director for education and a partner in Virtuous project. Today, they're gonna give us an overview of Virtuous project. We would like to apologize in advance if the video that will be shown may appear in low quality or grainier than usual. This can happen due to the unprecedented surge in internet traffic of these days, caused by many people using more and more the internet and virtual meeting platforms like this one we are using in the coronavirus situation. I hope the quality of the internet connection does not prevent us from enjoying the webinar. In any case, all the extracts and the videos that are being presented can be found on Canvas platform so that you're able to watch them again or in an extended version. I would like to remind you that your microphones are by default on mute and you're not able to start your video. If you have any question regarding the content of this webinar, you can post them in the discussion forum of Module 1 on Canvas platform, and the presenter will be more than happy to answer your question during the following days. Now, well, I'll leave the floor to you, Susan, Michael, and Adam, and see you later.
Thank you.
Thank you very much, Tekla.
Thank you.
So, welcome to our MOOC. This is a VirtuRack, and this is what we're going to talk about today. So the Virtuous project, it stands for Virtual Interactive Resources and Tools in Universal Education of the Sea. And this project has 5 scientific partners and 3 school partners from Sweden, Spain, Germany, and the US. This webinar that you're watching today has been designed by myself, Michael, and Adam.
And we also have a lot of users around the globe. Thanks a lot to this MOOC. For the moment, there are participants in the Virtue project from 22 countries. You can see all the flags here. Hopefully there will be even more joining us.
And we are pleased to say that we have over 350 registered participants for our MOOC, and we'd like to find out a little bit about who you are. So what we will do is we will have you participate in a poll momentarily. At the conclusion of the poll, we will see the results on our screen. We'll be asking you about the following types of questions related to your profession. Are you a school teacher at the primary level, secondary level, an informal educator, or another category? Thank you for participating.
You have another 15 seconds to place your vote. So you can see that we have many from the informal sector and also other, and we have 10% school teachers, secondary, and some primary school teachers as well.
You can also see that there are some, say, for example, Diana, that she covers all levels in her teaching ambitions.
So the MOOC is composed of 4 different webinars. The first one you're watching today, that's the introduction of the VirtuS. Then we have part 2, marine biofouling and biodiversity, on the 30th of April. After that, we have ocean literacy on the 7th of May, and the 4th one, how to organize hands-on activities on VirtuS, and that's on the 14th of May. So now we will show you a video which introduces the basic concept of the Virtu project. There will be a short pause where we share the video. Enjoy!
Hi, I'm Michael Olsson from the University of Gothenburg, one of the partners in the VirtuS project.
And I'm Adam Frederick from the University of Maryland Sea Grant College program, partner in the VirtuS project from the United States.
And now we want to welcome you to our MOOC.
Our MOOC will be promoting ocean literacy and project-based learning in the classroom by monitoring marine biodiversity.
This is a Virtu rack which we use when we are monitoring marine biodiversity.
When the rack is placed into the water, organisms colonize the surface of these discs. Why don't we go get one that's already colonized?
Yeah, we'll do that.
Okay, let's go.
So this is where we have placed our racks, just close to the Kristineberg Marine Research Station, which is just close to us here. And now have a look and see what we can find. And please note
yes, look at this.
The importance of a sign saying who has deployed it when, and some kind of reference to contact information.
Excellent. Let's take a look at one.
Yeah.
Oh, look at all this algae growth. Wow, look at this. Look at all these sea stars up here.
Yeah, the sea stars, they are predators on some of the creatures.
Oh, and look underneath.
Oh, they have left something to us. Wow.
We have bryozoans and tunicates, and more bryozoans here, a couple different kinds. Worms. Wow, these look fantastic.
Yeah, really.
Oh, look at that. There are some small barnacles on here.
Yeah.
These are going to look great in the laboratory.
Yeah, really.
Here you see a picture of the different rack parts. The Virtu rack parts can be bought in most hardware stores, except the discs, which are made of 3mm plastic, could be PETG or acrylic. These can be manufactured by plastic manufacturers. The ID tags can also be made out of plastic. Use a soft lead pencil to write on the ID tags. And not least, clean and reuse the plastic part except rope and cable ties. The ocean does not need more plastic particles. The University of Gothenburg is a Virtu hub for Sweden. This means we send out rack parts to Swedish schools without charge. We are hoping to have more Virtu hubs around the globe. And if you are interested in being a hub for your country, please contact us. You can also add petri dishes, bottles, pieces of wood, etc. to do biofouling studies on other type of material.
Here you will watch a video on how to mount a VirtuRack. Here you will watch a video on finding the best place for a VirtuRacks. So racks can be deployed in freshwater, brackish water, or seawater. Please enjoy this video.
There are some field-related factors to take in consideration. For example, transportation. Virtue discs can be transported, for example, in sealed thick plastic bags with some kind of moisture-preserving material like wet newspaper. Or you can transport them in a simple cooler with a tight-fitting lid. Suitable bags can be purchased in aquarium stores and hardware stores. And the air inside the bags will now have 100% humidity, and the animals can do well with this oxygen supply. Close the bags with a cable tie or a rubber strap, and keep the bags cold in a refrigerator if you have the possibility. That will increase the survival time. Place some kind of spacers between the discs if you have dismantled your rack. You can transport it without dismantle it, but if so, you can minimize the risk of squeezing the animals. And remember to also bring water from the test site. You will need this water when you do your disc observations. In the investigation, if the investigation does not occur immediately, use water from the test site and replace the water in your boxes with the discs each day or after the weekend. Provide aeration from, for example, an aquarium pump, if keeping the discs for an observation period more than about 24 hours. Also, do not forget to open the covers when you get back to the classroom, and place the buckets where you keep the discs in a cool, shaded place. Other field-related factors is the familiarity of the site and the location of the site. Is it from a dock, a pier, a jetty? It could be a stream, a pond, a lake, etc. And the safety of the location. The disc material could, as I said before, be made out of acrylic or PETG, but you can also use other kind of material. And we got a question in the forum. Why are we using a pair of discs? That's because when in the lab or when you're observing the discs under a microscope, you can observe the upper side, which is more exposed to the sun and the light, but is also having more sediment. And on the other hand, the lower disc of the pair has less light on it and less sediment. So that will most likely result in different fouling on the discs. And also the overall water quality and the flow rate of the water or the tidal influence if applicable. And the depth of the rack in the water. We recommend 50 centimeters under the surface for the upper pair of the discs. Seasonal influences, leaving the rack in the water during, for example, at least in the north of Europe or America, cold and winter time. We also got a question for how long time should these racks and discs be deployed, and that's really related to where you have deployed it. Let's say 2 to 4 or even 5 months, but you can also observe them after 1 year and see the difference related to those who have been newly deployed. It's up to you as teacher to decide. Seasonal influences, the length of time to leave the rack in the water, as I said, and accessibility and prevention of tampering, that is quite rare, fortunately, but it happens.
So this is the VirtueS website. You can see the exact address up in the left corner. So this project provides basic marine species identification guides. You can also use our database to register what you have found and also to register your geographic location. You can upload images, you can blog about the school's project, and you can also find out about other projects and their geographic locations. On the website, we also have different reporting forms in PDF, for example, that you can download. And on these, you can record the organisms that you find, and you can do this while you are in the field or in the lab. We also have an online tutorial for random sampling to assist with counting of organisms, and also a tutorial on key measures of biodiversity. And that tool can be used to refine the raw data from counting on the disk. We have, if you want more details on how to use the website and other useful things, please watch our video capsules. We will show them in a second. We also have a search function on the website and a sitemap, and this we've added to make it easier for you to find things on our website. So these are the video capsules that I mentioned. So you see the titles here, and then if you will go into the Canvas portal, the course portal, and check under module 1, you will find all these video capsules to watch. In addition to biodiversity data, teachers and students are also encouraged to record temperature, pH, salinity, water transparency, and other variables. And not only should these data be collected when discs are immersed into the water and raised from the water, but these measurements can also be recorded several times in the intervening period. So now we would like to play this video for you, VirtuRack in water, retrieved and observed.
The VirtuRack is now safely retrieved from the seawater and can be observed on the jetty as it is or dismantled for further observations in the lab. Note the green algae, leaf-like. There are several species here to be found. And there is a lot of barnacles, both living and dead. And here is a blue mussel, a small one, 1 year old about. And here is an oyster, Crassostrea gigas. Now take photos of the discs as close as you can, or bring it to the lab for further observations.
This is what it can look like under your microscope. Of course, it differs. This is from the west coast of Sweden. You will find other species, of course, in the Mediterranean or outside Charleston, etc., etc. if you only want to study what's growing on the discs at different times of the year, all this can be observed at the same time. Or if you want to follow the changes in the composition of the species over time, retrieve some discs and let others remain in the water.
So the Virtue project requires some planning ahead. At schools, the project have to be synchronized with the timetable. And Virtue discs, as Michael mentioned before, need to be in the water for several weeks or even months, depending on season or your geographical region. Here we present a time frame to give schools an idea on how to fit Virtue into their schedule. Something to remember is consider the environment to be studied and perform some research about the area. This will give you background information on the aquatic system and factors that may influence the biodiversity. This type of background will help guide the development of your research question. First, we recommend that you become a member on our website. And then you can start sourcing for rack parts locally. In Sweden, we mostly deploy our racks in April and then start the analysis in September. But of course, that depends on what you are doing for sort of studies. After the analysis, you should clean your reusable rack parts and have them ready for your next experiment. You should have a research question ready before you begin. Have a look at our website for ideas for research questions. Virtue provides many opportunities to take an investigative approach to one's work, practice in setting up experiments and reporting on the results. But you can also use video as a tool for documenting your findings, which emphasizes the integration of humanities in the sciences. Our project partner, Dennis Augustsson from University of West in Sweden, will talk more about that in this video that we are showing you in a second. We will not show you the whole video now, but you can watch it later. It's on Canvas under module 1. Dennis's video also includes 3 videos made by high school students that are part of this project.
Hi, my name is Dennis Augustsson and I'm a researcher from University West in Sweden. I've been training our partners and their students in media literacy and video production in the Virtue project. And why have we done that? Well, video production gives the possibility for teachers and students to produce and present knowledge in a new and creative way. By documenting and sharing their experiments, they get a deeper understanding and the possibility to connect to other schools and discuss and share their experiments and results. This is the basic structure for our collaboration. First, the students met in a video conference to get to know each other and to discuss Virtue topics. After that, each school produced videos on their own Virtue experiments and then published them online for the others to watch. After that, a second video conference was set up, where students discussed their results and processes. Making videos on your science experiments is not mainly a question about technology. You can use your own smartphones and simple editing tools available for free on the internet. But what you have to learn and discuss with your students properly is how to make a good narrative on video. What scenes to shoot, what kind of images to use, and how you explain the experiment and the results in a comprehensible way using video. Now, this is a skill that takes some time to practice, but it's a very rewarding and creative process. a good advice is to make it a cross-curricular activity using English, fine arts, or other subjects that could be part of the filmmaking process.
In our next slide, we're looking at analysis, imaging, and microscopy. And we will show you a video shortly that describes some of the methods being used there. Some of the important things are to use a very good type of light. There are many different varieties of this when you're looking at images through the microscope and analyzing your discs. Some of those include LED lights, which are very common now and provide a good white light for observation under a stereoscope. And others include gooseneck lamps and other types of fine point lighting for microscopy. It's important to understand the context which to set up the observation skills for the students. What we recommend is that students should use their naked eye first for observational skills to see what they can pick out on the disc, colors and textures and movement among the organisms, and then progress to something like a magnifying glass where they can make observations up close and make more descriptions and even drawings on some notebook or journal, and then move on to the microscope. So they'll have a context for what they'll be looking for at much closer range and much closer focus.
So the type of analysis that students can do, well, they can choose to only identify taxonomic groups or according to their feeding strategies. But they may also investigate how the organisms adhere themselves to the disc and if there is additional fouling on already existing fouling. Or they can investigate the effect of anti-fouling paint on the discs. And that is actually one student group is doing that. You can, if you watch the complete version of Dennis's video, you will find that student video there. They can also estimate biodiversity on the discs. Have a look at our microscopy video where Adam will talk about what equipment to use to capture images. And let me remind you again to watch this video in full on Canvas.
Wow. This morning we're going to show you some of the technologies used to connect cameras or video cameras, your phone camera to the microscope. Most of you are familiar with a stereoscope or other type of microscope, and the first thing you need to do is remove the eyepiece. So that's the key. So most microscope eyepieces are either 30 millimeter or 23 millimeter. So if you're using one that is smaller than this one, then you would unthread this piece of the adapter and thread this one in, and you can put it into a smaller eyepiece here. Or the port for the camera on the microscope. Sometimes there's a third port on the microscope called a C-mount, and you can put your camera in there and still have your two eyepieces here. But most of the time, there isn't a separate camera port on your microscope, so you just take the eyepiece out. So I'll put this one back in. And then video cameras, as you know, have gotten better and better and better as time has gone on here. But some things have changed about video cameras. If you're going to connect a video camera to this adapter, you'll notice it has threads on it that thread into the lens of the camera. So most of these video cameras now don't have the threads here, which is called the filter diameter. And if you're doing any photography work or have cameras at home, Nikon or Canon or whatever, sometimes they have threads here and sometimes they don't. So the newer cameras now don't have these threads. So you may have to look for an older camera to go with this adapter, or you rig it so that it connects together. but these both have matching threads, so this one can just thread right onto the camera here, like this, and then plug this right in here. And just open that up, and then you have the ability to see what's in the microscope. So at first, when you bring up this camera image, you may see just a circle of light, so you have to zoom in to get it to fill the screen. So this afternoon, when we're doing our sampling and looking at the live samples under the scope, I'll show you exactly how that works. But this is an older system. That's how this camera system works right here. And when you take it out, you can kind of see the circle there. All right, because you have this vignette here. This is much smaller than the lens of the camera. So to get rid of that, you would have to zoom in like this to fill the screen, like that. So that would give you the image under the scope then. So you're kind of using more than the magnification of the microscope. So it's hard to get an exact estimate of what the size of the organism is unless you measure everything ahead of time and you have a standard. So when you zoom in, you know how big the organism is. Because you're zooming in farther than what the microscope is doing at low power, let's say. So if you zoom back out, that's not the picture that you want. You don't want that little circle there. You just want to fill the screen. Then you move on to more, today's technology, where here is your camera, right? So the advantage here is that most everybody has a phone, and most students have a phone. So one of the newer ones, and I can pass this around so you can see it. So this company develops adapters for the iPhone. I don't think he's doing Android yet, but this is the adapter using the same principles as the one I just showed you. So here is the adapter for this specific size phone. So this adapter fits an iPhone SE. That's this model right here. My phone's probably about 5 years old at this point. But I keep it because it fits this adapter really well. So the camera just lines up with the adapter and just sits right inside there, like that. And then it's the same principle. How do we get connected to the microscope? So same technology. He has a 30 millimeter adapter or a 23 millimeter adapter. So same thing. You would just thread this in to here, and then it slides into the microscope eyepiece, but you would want to remove this eyepiece. So now we're going to consider some of the teacher perspectives and the benefits of the Virtue project and 21st century learning skills in the next few slides that you see here. Virtue is a school project aimed at stimulating an interest in science among students and also to contribute to further development of teachers. Virtue aligns well with primary and secondary learning objectives, things like formulating questions and interpreting results. In both the US and Sweden and Spain and Germany from our project, we've learned this through the teachers we've been working with. And most likely in more countries that Michael has mentioned before at the beginning. At older ages, Virtue can be used as an interdisciplinary approach in biology, mathematics, environmental knowledge, and physics. Virtue is also suitable for thematic projects. We're living and working in an interconnected and globalized world. Therefore, it's essential to prepare students to enter into this world, and they will need to be equipped differently than past generations. Students of today must be able to collect, analyze information, do research, and be part of the team. At the same time, students need to respond to other demands, such as advancing technologies and new global requirements. The 21st century learning skills of collaboration, communication, critical thinking, and creativity all come into play in the Virtue Project. It offers opportunities for students to participate in international collaborative learning experiences, as you will see in the next few slides. This will engage students, improve their intercultural awareness, personal development, and cross-curricular skills, and give them opportunities to discuss global environmental issues, things like biodiversity, climate change, and ocean pollution. Here are some various pictures that show our Virtue Project. This one is from the Baltimore Harbor in Maryland, where I work. And there are other photos here that include the Grice Marine Laboratory, as you see here in Charleston, South Carolina. These are a group of Swedish students working alongside peers from James Island Charter High School in Charleston, not far from the lab. And this is a very unique opportunity since the Swedish students sail into the Atlantic and one of their stops is Charleston. So we've set up this partnership and been running this program for about 3 years, where the local students are meeting their peers in Sweden and working on this biofilms, biodiversity, and virtue project together. And then they can also share their information and video conference with one another, which you will see when you watch the complete video that Dennis has posted about media literacy. So this opportunity became very popular with the local school and the Swedish students, and it's our hope that we will continue this every year when the Swedish students do visit. And here is a picture of all of them outside the Grice Marine Lab at the College of Charleston in South Carolina. This is one of the discs that we analyzed, and it offers an opportunity for students from both Charleston and Sweden to look at the great biodiversity in the Charleston Harbor. And this disc was probably in the water for about 3 to 6 months. This next slide, we're looking at 2 aspects of the Virtue Project, which we consider to be important parts. And one of those is called project-based learning, and the other is called a 5E instructional model. So in project-based learning, students are basically driving the activity and are engaged in real-world and personal, meaningful projects. And this is important for students to be leaders or co-leaders along with their teacher, and for the teacher to be a facilitator in their learning. In the 5E instructional model, this is something that came out of research in the 60s and 70s in the US and came from something called the learning cycle. It was then further expanded into a 5E model in the early 1980s. And this includes these 5E parts, starting with engagement, and then that leads to some exploration by the students about the engaging topic. It leads them to further explain their exploration and their results, and then to elaborate that further and connect it to local society or local issues where they set up their project. So their local harbor, for example, and pollution issues surrounding that. And then it also provides an opportunity for the teacher of the students to evaluate them and how they design their project, how they present it, and in this case, the videos that they constructed. And also it provides an opportunity for students to do some peer-to-peer evaluation of one another in a way that creates a very interactive environment for the students. We really think that Virtue, with these aspects of project-based learning and the 5E, create an excellent model.
So now we are wondering, have you heard of PBL and the 5E before? We are doing another poll, so I'm just going to get it going here. So you can answer yes, both, only PBL, only 5E model, or none of them. So now you have 20 seconds more to vote. Okay, 30 seconds are up, so let's end the poll and share the results. So you can see that there is a mix here. 90% have they know of both. Some 41% only PBL, and 9% only 5E model, and 31% has answered none. So let's move on. We have another question for you and another poll. What methods do you use to teach science at your school or educational center? You should be able to see the questions now. The question now. What methods do you use to teach science at your school educational center? You can choose between PBL, 5E model, both PBL and 5E model, lecture-based or other. You have another 10 seconds to place your vote. Okay. Now I'll share the results with you. So we can see that the top two ones, we have 31% each for PBL and lecture-based. We have 3% on 5E model, another 7% for both PBL and 5E, and then we have 28% for others. Now we have another video for you to watch. This video is with Adam again, and he's talking about the 5E model.
The pedagogical part that relates to the Virtue Project. I think the Virtue Project does a great job using the 5E components, and they would get a thumbs up from Leon Letterman. So, well, you just weave it in yourself. It doesn't have to be an outward thing. And nobody's saying that all those E's have to happen in one class period. That's the other thing I wanted to mention. Nobody's saying you have to do that in 45 minutes or an hour. This could take, depending on what you're doing, 2 or 3 days. Or you could do something very quick that used all of those in half an hour, depending on what the activity was. So it really is up to an experienced educator to sift through all of this and make it happen. But experienced educators don't happen in 2 or 3 years. Just a note, we have a few 5E models to follow, so if you wanted to see how they're constructed, we have some here. Well, this is one that we did a few years ago called Fish Oil Really. It's the whole idea that omega-3 oils aren't made by fish, they're made by algae, and they're accumulated by fish in the food chain, but most people don't understand that idea. So we take them through a whole series of activities so they can get the whole idea behind that in a 5E type format, right here. So you can click on each one of these and it goes through the whole activity with you, but that's what it looks like on the web. So we have a few models there for people to follow. Yeah. So in this slide, we're trying to weave together the ideas that we've mentioned briefly here about the Virtue Project, project-based learning, and the 5E. And some of the important points here are that the Virtue Project focuses on student-driven inquiry and observations, as we've talked about. It makes it easier for students to see the connections between what they observe and theoretical models. It has been shown that students taught through field work and classwork have become more proficient in that particular topic in comparison to those taught without some field work experience. It is important for students to have time to reflect on their experiences in the field, as it will make their understanding more profound and make them realize its relevance to the world outside the school. Virtue provides the opportunity for students and teachers to study various parts of the ocean or freshwater environments, and this promotes the understanding of interconnectedness among the different geographic locations. So as you can see in the photos we showed before of the students between Charleston and Sweden, that creates a really well-focused example of what we're discussing here. And the way that they contact one another and interact with one another through video conference enhances that part of the study that they can connect to on the web and through the data that is recorded and observed. For students, it's really important for studying both the water quality and the biodiversity to enhance perspectives about human impacts on aquatic ecosystems in their local environment and how that's connected within their school classroom.
So now at the end, you have assignments to do. There are 3 questions and you will find them on the course module portal at Canvas. So have a check there and follow these instructions. You can also ask your questions in the Canvas forum for each module, not only module 1, as Tekla said at the beginning of this webinar. There has also been some questions during this webinar. Some of them have been asked, has been answered, but please place your questions in the forum so everyone can see both your smart questions and hopefully our smart answers to them. And if you are looking at this in the recorded version, you can still use the forum if you have further questions. And please have a check, maybe your questions have been answered before. Hopefully there will be a lot of questions, but it's easier for all of us if they are not repeated too many times. So thank you very much for listening and watching, and remember that the videos can be looked from the Canvas in better resolution, hopefully. So over to our moderator, Tekla.
Thank you, Michael, Adam, and Susan for the talk. I hope that everybody enjoyed it. And as we are saying and as we are repeating, please do not hesitate to post your question to the presenters in the forum of discussion. So they will be answered in the next days, and maybe, as Michael said, those questions can help also other participants. And we will post the recording of this webinar on Canvas if you would like to watch it again. And I would like to remind you, as already said by Michael, that at the end of each module, you need to submit an assignment that you can find on Canvas platform under the section assignment. And once you have submitted all the assignments, we will issue you the certificate of participation to this massive open online course. And save the date for the next webinar that will take place next Thursday, the 30th of April at 6 p.m. Central European Summertime. We will talk about how to study biodiversity on Virtuous Disc with Professor Sally Deng. She's from Geomar Helmholtz Center for Ocean Research based in Kiel, Germany. And the Geomar Helmholtz Center is partner of Virtuous Project too. Thanks again for attending today's webinar, and stay safe and take care. Bye-bye. See you.
Bye-bye.
Bye-bye. Thank you.